So we're gonna talk about the study guide. I'm gonna give you a general idea what I want you to know. It's not gonna be very much different from the study guide recording that we have seen, before. If you have any questions during the recording, please use the chat to let me know. That's unfortunately, not unfortunately, that's probably the best way to communicate with me during recording. So bacterial cell division, binary fission. So what you need to understand about bacterial cell division in general. So it's binary fission.
Sorry.
Yes.
The chat isn't up on my computer. I don't know if it's up on everyone else's, but I don't have the option.
Try to move the cursor over at the bottom of your screen.
Yeah. Oh, it's just not enabled. Okay, sorry.
Yes,
Okay.
because when somebody writes something in the chat, it should pop on everybody's screen. I can actually open the chat window here. I don't know, Darla, you will be my responder. Can you see the chat window on your computers? Okay, good. So if you have a message or a question, you can write it down here, either for everyone or specifically for me. So cell division is binary fission. Bacterial DNA and cellular components have to be doubled. Well, cellular components have to be replicated, and DNA definitely has to be replicated. And remember that one important protein that has to be expressed is the FTSZ protein that serves as the a tie that separates two cells. When they divide, it forms the cleavage furrow between two mother and daughter bacterial cells. Number of bacterial cells actually change after each division. Doubles, generation time is the doubling time. Remember we discussed the generation time, where it is longer, in the lab or in the natural environment. Natural generation time is always longer because the conditions are better, and it varies between the bacterial species. Okay, so basically generation time depends on the species, on the conditions like pH or temperature, and whether or not there's competition, and so on and so forth. So these two, you should be able to calculate the generation time based on the number of generations, initial and final number of cells. The example in the questions that I provided gives you an idea how to do this, for how to calculate the generation time. I also have, we also had a couple of questions in the quiz that you took before the spring break that was explained number of cells, so I expect you to know that. Growth curve, okay? So you need to understand the steps of the growth curve, okay? X-axis versus Y-axis. And the main phases of the growth curve. Now, what I mean by the main phases. What's in the lag phase? Number dividing cells. There are no dividing cells, no dying cells. Nutrients are not used, wastes are not produced. The logarithmic phase, number of dividing cells exponentially increases, number of dying cells is minimal, nutrients are abundant, wastes are virtually non-existent. Is that a highlighter? Yeah. Number of dividing stationary phase, number of dividing cells, it's pretty much equal to the number of dying cells. Nutrients and wastes are pretty much balanced. Okay, my window, can you still see me? Okay, the screen, I mean, that's most important. Okay, good. Decline phase, number of dividing cells is practically not there. Number of dying cells is overwhelmingly high. Nutrients are practically gone. Wastes are very abundant. Persister cells, by the way, you probably know there's fill and sign option in Adobe Acrobat, which basically allows you to type stuff into your document, which is kind of cool, I think. And you can change the size of the font and stuff like that, okay? Persister cells, cells that are formed during the decline phase, and they have reduced metabolism. Pretty much reduced, very much reduced metabolism. Hemostat is the device that allows to maintain population of microorganisms in the logarithmic phase, okay? Now the direct cell count, those are methods for counting cells. You should be able to compare and contrast them. Direct cell count, use hemocytometer. You usually count the total number of cells, but you can tell the live and dead cells apart. The advantages, of course, I listed in the exemplary question. That it's fast, cheap, simple. Disadvantages, it's tedious, and it's subject to operator bias because the person actually counts them. So Coulter counter method does not have that disadvantage of the operator bias, but you cannot tell live and dead cells apart. So the advantage is that it's fast, automated. Disadvantage, well, cannot tell live and dead cells apart. And also I want to remind you, you have to work with the very diluted solutions of the cells. Viable cell count is the gold standard. You have to prepare serial dilutions, and usually you dilute by the factor of 10, and then you use either pour plate or spread plate method to plate those dilutions and calculate the number of the viable cells. Remember, it's very tedious and time-consuming and effort-consuming work. Filtration, how can it be assessed? I got a question about this yesterday. So filtration, let's say you want to sample water from a nearby river. If you just take 100 microliter sample and put it on the plate and spread it on the TSA, most likely you're not going to see any growth because the sample is extremely diluted. Okay, but when you swim in that river, you're not accidentally consuming 200 microliters. You may gulp in a pretty good chunk of water. So you need that good chunk of water, say a liter or 2 liters. You can collect that volume, filter it through a filter, and then everything that's in water, I mean bacteria obviously, because you want to remove all the sediments, bacteria, they remain in the filter. You can either put the filter in the special medium or do a replica plating on the TSA or any other plate that you want. That's kind of an idea. So that's about filtration. We got it. Most probable number. I know that it's a bit convoluted. I am not so much focusing on the mechanics of the method. You know, 5 tubes with 10 mils, 5 tubes with 1 mil, 5 tubes with 5 milliliter aliquots. I am more focused on you understanding what type of information we can get from that method. Do you get an exact, so like you analyze diluted samples, and does it give you an exact number? No, it doesn't. It gives you a probability of a certain number of cells. It does measure live cells, only live cells, not total, live cells. And it gives you a probability, it gives you a number that is the highest probability. So say 57 cells, that's the most probable number, okay? You have highest probability that your sample has 57 cells. Can it, in actuality, have more or less? Yes, it can, absolutely. But the highest probability is 57. So, and it also gives you the interval, the confidence interval. So turbidimetry, measuring optical density. So you're not measuring the number of cells. Understand that, please. What you can do, remember we were talking about assessing them, using turbidimetry as a really, really fast, convenient, quantitative method to build the growth curve. If you do once an actual growth curve using serial dilutions, okay, and you run turbidimetry along with that, you can match optical density with the certain number of live cells. So you can basically say, okay, I've got, I can actually, I think it's pretty cool to say, I can say, okay, 1,000 colony forming units per mil equals optical density of 0.4, okay? And you can run two graphs, okay? You can run two graphs here, right? One graph represents the actual number of cells, another graph represents the optical density. So we measure optical density. This parameter becomes higher in the concentrated suspensions. In which macromolecules you can find oxygen, carbon, and hydrogen? In all biological macromolecules, actually. Nitrogen. So remember, it's a DNA and proteins. Phosphorus. Somebody pointed out that phosphorus is found in both DNA and DNA and RNA, DNA, RNA, and sometimes proteins. Same is true for nitrogen, DNA, RNA, proteins. Sulfur is practically exclusively proteins. Sulfur roles for potassium, sodium, calcium. Potassium, sodium, and calcium establish the membrane potential as well as chloride. Iron, magnesium are two enzymatic cofactors. Nutrient, chemical without which microorganism cannot grow. Organisms that oxidize reduced organic carbon. Those are heterotrophic organisms in the process of cellular respiration. Photosynthetic autotrophic organisms reduce oxidized inorganic carbon in the process of photosynthesis. It's all in the answers. Kind of describe how you should answer those types of questions. Methanogens use carbon dioxide as the final electron acceptor. And they, as the result of this reaction, they produce methane, thus contributing, methanogens contribute to the greenhouse gas emissions. Okay. Methanotrophs, they use CH4 as the source of carbon, and they convert CH4 or methane. Remember, CH4 and methane are the same. They convert it into various organic compounds. So methanogens, they contribute to the greenhouse gas emissions, and methanotrophs, they reduce greenhouse gas emissions. So atmospheric nitrogen, when it's reduced to the ammonia, call it ammonification. That allows living organisms to incorporate ammonia into the biological macromolecules. I'm just checking that it's recording. And then ammonia is oxidized to nitrites and nitrates. This is nitrification. And nitrification also increases the motility of ammonia and provides the final electron acceptor for some anaerobic respiring microorganisms. And then when that is a process of nitrification, and nitrates, when they are reduced to molecular nitrogen, this is denitrification. And the practical application, the denitrification, is to the wastewater treatment. When you have agricultural runoff that contains various nitrites and nitrates that are used as the fertilizers, you can neutralize those fertilizers by basically reducing them to molecular nitrogen, denitrifying. I'm going to take a little pause here to remind everyone, if you have a question, you can just type in the chat like a question, and I will stop and I will wait for your question to appear, okay? Hydrogen sulfide, it can be converted into sulfates by photosynthetic bacteria. And sulfate ion is used as the final electron acceptor in the process of anaerobic respiration, which leads to the reduction of sulfur into hydrogen sulfide again, and reduced sulfur is essential for living organisms to incorporate it into various proteins. Now, symbiotic relationships, mutualism, obligatory relationships in which both microorganisms benefit. Commensalism, mandatory, obligatory relationship in which one microorganism benefits and another microorganism doesn't. Nothing happens to it. Parasitism, one microorganism or organism benefits and another organism suffers. So it's one organism thrives at the expense of another. Difference between ecto and endoparasite. Ectoparasite is living on the outside of the body, and the parasite lives inside. Obligate parasite, the one that must have a host, must parasitize some other organism, and intracellular parasites are the ones that can only survive inside the cell. Satellitism is an example of the usually nutritional type of relationships. Satellitism is an example of commensal relationships when one microorganism produces an essential nutrient for another microorganism. Non-symbiotic relationships, synergism and antagonism. I think those two were examples on the practice questions. So synergism, microorganisms can live without each other, but when they're together, their growth rates are considerably higher. And antagonism, when microorganisms they compete, and one destroys another, kills it. So you can see the difference between parasitism, in which parasite doesn't kill the host, and antagonism, when one microorganism kills another. So planktonic growth, growth in suspension, biofilm on the surface, biofilm is more common. Biofilm formation starts with the primary colonizers that form extracellular polymeric substances, and their role in the biofilm is to provide a matrix for the biofilm formation, provide that sticky goo that attracts secondary colonizers. So basically the difference between the primary and the secondary colonizers, primary colonizers stick to the surface first and produce extracellular polymeric substance. Now, extracellular polymeric substance, along with being goo, so protects the members of the biofilm against the desiccation, predation. Critical density is the amount of microorganisms at which members of the biofilm start to produce quorum-sensing molecules, regulating the density of the biofilm. Okay. Okay. So species in the biofilm usually commensal or mutualistic relationships. Okay. So they are definitely symbiotic. So we are talking about commensal or mutualism. I know what's going on there. The great thing about PDF, you can actually annotate it very conveniently. If you use computer, it's really, really good. Gram-positive microorganisms, the quorum-sensing molecules are short peptides, and gram-negatives, large organic molecules, called the lactones. Homoserine lactone, for example, they're different because gram-positive and gram-negative microorganisms have different structure of the cell wall, and they have to use different, different molecules have different way to cross the corresponding cell walls. Quorum-sensing molecules are autoinducers. It means that they can stimulate their own production. Aerobic versus anaerobic. Aerobic needs oxygen, and anaerobe does not need oxygen. So obligate anaerobes, oxygen requirements. It cannot survive in the presence of oxygen and will most likely be fermenting microorganism or carrying out anaerobic respiration. Okay, either fermenting or respiring anaerobically. For obligate aerobe, it must have oxygen and it will have normal aerobic respiration metabolism. Requirements and metabolism for facultative anaerobe. So it grows best in the presence of oxygen, but can also carry out fermentation and grow without it. This is why we call it a facultative anaerobe. The metabolism will be cellular respiration. Oxygen requirements and metabolism for air-tolerant anaerobe. It doesn't need oxygen, but it can grow in the presence of oxygen. And metabolically, it is usually either a fermenter or anaerobically respiring microorganism. Microaerophiles, they need oxygen, but relatively low concentration of it. Okay. Anyone has a comment? If you have a comment, you can unmute yourself. You will go on the record though, but you can unmute yourself and say something. Okay, microaerophiles, they need oxygen, but in concentrations lower than what we call, regular, normal. And this is why we call them Goldilocks, because they can grow at this exact concentration of oxygen. Catalase, peroxidase, superoxide dismutase, they neutralize reactive oxygen species. Specifically, catalase and peroxidase neutralize hydrogen peroxide, superoxide dismutase neutralize superoxide anion. So these enzymes, whether or not these enzymes are present, that will determine whether or not a particular microorganism can survive in the presence of oxygen. So basically, if neither of these enzymes is present, that will be obligate anaerobe. Obligate anaerobes die because they don't have these enzymes. On the other hand, firing microorganisms, obligate aerobes, they have all three because they are exposed to a lot of oxygen. They're running respiration that generates a lot of reactive oxygen species, and they need all three of these enzymes to neutralize the generated reactive oxygen species. So which obligate anaerobes? Air-tolerant anaerobes usually have peroxidase. They may have catalase, but mostly it's peroxidase neutralizing hydrogen peroxide. Okay. They don't have superoxide dismutase because for them, since they don't really, let me start over. No. Tolerant anaerobes. Let me actually type it. I can do that. Yeah, I can do that. That's pretty cool. So do not have obligate anaerobes, to clarify. Air-tolerant anaerobes, they have SOD, superoxide dismutase. Facultative, they have SOD and catalase. Air-tolerant anaerobes, so catalase, basically, the answer here is catalase is in facultative anaerobes, not in air-tolerant anaerobes. It can be used in bacterial diagnostics because you can basically run this catalase test. You take a sample of your bacteria and you put it in a hydrogen peroxide. If bubbles are produced, there you go. That's your oxygen is released because of the catalase. Hapnophiles, microorganisms that require carbon dioxide for the growth. Growth pH, come on, people. Acidophiles, neutrophiles, alkaliphiles. You got to remember your pH. Pathogens, human pathogens are in the neutrophile group, 5 main groups of bacteria based on optimal growth temperature. Will I require, so let me explain. The advantage here, I mean, I can give you numbers, like I think I did some, well, anyway, let's say I tell you, we have a microbe that grows at 100 degrees Celsius. And you respond to me, oh, that's a thermophile. Will I be really PC about the fact that you didn't say extreme thermophile? No. Thermophile, extreme thermophile, it's fine. Like, to be on the safe side, just tell me that this is thermophilic bacteria. Tell you there's a microbe going at 0 degrees Celsius. You tell me, oh, that's psychrophile. So believe me, I'm not gonna throw you under the bus and give you numbers that are borderline and you will be anxiously figuring out what you're supposed to say. It's gonna be pretty obvious, okay? Now for psychotrophs, remember, they are of particular interest for the food microbiology because they can grow in the fridge, basically, and spoil the food. Human pathogens are mesophiles. Osmophiles are microorganisms that can grow in the presence of solutes, high concentrations of solutes, any solutes, sugar, salt, proteins, doesn't matter. Halophiles is the subclass of osmophiles that can grow in the presence of high concentration of salt. That's the only difference. Halophiles a bit specific. Barophiles, high pressure, usually at the bottom of the ocean. Xerophiles, dry conditions. Polyextremophiles, multiple sort of resistance, resistance to acid, radiation, desiccation, temperature, you name it. So that's basically multiply resistant. Methods of microbial control based on the object being treated. Methods that are applicable to treat fomites, that are applicable to treat patients, aseptics. Cleanliness level. Know that stuff. Sterilization versus disinfection versus sanitization for fomites, asepsis and degermation for patients. Since you're all muted, you clear? Everybody's good about that? Like, that's really straightforward. Question? Anyone has a question? Seriously, type it in the chat. I can unmute you, okay? And you can ask me a question.
I have a question.
Introduce yourself. Because I have...
Rosalba.
Oh, Rosalba, hey, how you doing? Yes, go ahead.
So, like, what exactly is... I guess I'm having trouble distinguishing between disinfection and sanitation.
So let me rephrase it, okay? And if I understood you right, you just say yes. You know, what is the difference between disinfection and sanitization?
Yes.
Good. So in case of disinfection, are using a particular method that would destroy the cells via chemical approach or physical approach. Let's say when you pasteurize, you use temperature to kill, like physically kill microbes. When you sanitize, it's more of a mechanical removal to reduce the number of microorganisms by, yeah, you can use the chemical like soap. Does that make sense? But soap doesn't really kill microorganisms. It just removes them. Or let's say you have a dirty dish and you wash it under water without anything. Will water remove microorganisms from the surface of the dish? Absolutely. But it's not like a specific action of temperature or specific action of a chemical. Did I answer your question?
So sanitation, there's a specific method, mechanical method, basically.
Disinfection, a chemical method or a physical method that specifically kills microorganisms. Usually not all of them, but the majority of them. In sanitization, it's more of a removal, mechanical removal. Does that make sense?
Yeah.
So it's like healing versus kicking out. Okay?
Okay.
Good?
Yes. Thank you.
No problem. Okay. Cleanliness level, type of the technique, that's what we just discussed. Main protocols for the control of microbial load on inanimate surfaces: sterilization, disinfection, sanitization, living tissue, asepsis or aseptic technique, and degermation. Levels of biosafety containment. It's a hard question because even in different countries, different pathogens will have different biosafety containment level. So if you feel comfortable with extreme, you're fine. Now, sort of a good and a bad news at the same time. If I ask you about something like part of the, let's say it's a part of the question, biosafety containment level for West Nile virus. I don't know. Is it in the United States? Yeah. Is it pretty bad? Yeah. Does it have a vaccine? No. Well, let's say let me rephrase it. something that we covered. Mycobacterium tuberculosis. Mycobacterium tuberculosis. What's the containment level? Okay, can it kill? Yeah. Is it in states? Yeah. Is it easily transmitted? Yes. Can you cure a person? You can. It's not super easy, but you can. So you say, you know, based on this data, I think it should be 3, at least 2, 3. 4 is probably an overkill. Like if you give me an argument, I can see an argument, and your answer, this is the advantage. Your answer may be a bit off, but if you provide a solid argument for your answer, like if you try to convince me that I can work with Mycobacterium tuberculosis on my kitchen counter, that's no, I can't. If you tell me that I can work with it using PPE in BSL-2, I can. I probably shouldn't, but I can, and I see where you are coming from, and you will still get a full credit. Does that make sense? Okay, I can only see Darla, so she nods, and I'm good. Okay. So geographical distribution. Basically, let me give you some details here. If the pathogen is exotic, and we're going to focus on the United States. If the pathogen is exotic, not present in the United States, it automatically puts it in BSL 3 or 4. If it's pathogenic, 2, 3, 4. The higher the virulence, the higher the BSL level. So if we're talking about something that cannot be cured and is going to kill you basically 100% of the time, yeah, it's BSL 4. If you have a vaccine, though, if you have a vaccine against it, that knocks it down a bit. So like rabies is going to kill you, but there's an extremely efficient vaccine and prophylaxis against it, and it's not easily transmitted, so it's surprisingly BSL too. So, you know, believe me, I'm not going to throw any examples that you don't know, or if I give you an example, I will provide you with an extensive explanation of like what this whole microorganism looks like. Most resistant endospores prions, least resistant sensitive cells, enveloped viruses. So that's basically that resistance diagram, I don't know, sequence that I gave you on the slide. Does that make sense so far? I hope I did. Sexual fungal spores and protozoan cysts are resistant to some disinfectant protocols, like in the middle. They are formed when cells really have to survive in harsh environmental conditions, so that's why they are resistant. Ranking, you're not going to be ranking. That I would do this if we would have a multiple choice, but not here. Decimal reduction time that is required to reduce the number of microbial cells 10 times. Lower means that either microorganism is more sensitive or the method is more efficient, and higher is vice versa. Cidal agents kill, static don't they just, you know, suppress the growth. Factors that can influence decimal reduction time: temperature, pH, pressure, presence or absence of organic compounds, overall load, the nature of microorganism. What you are treating, what's the outcome that you need, you know, whether you're treating, well, you know, living tissue versus fomites, what level of cleanliness is needed, time consumption, stuff like that. Main cellular structures, so cell wall, cell membrane, DNA, RNA, protein. Not going to be focusing too much on giving me the information. What pasteurization effects in the cell? Probably everything, you know. So don't get too hung up on these ones. Physical approaches, so temperature, radiation, filtration. Temperature mediated controls, moist heat, dry heat. Moist is more effective. Microbes rank exactly the same way in resistance to them. Thermal death time that is required to destroy all microbial life. at the given temperature, terminal death point, time that's required to, not time, sorry, temperature at which all microorganisms are dead in 10 minutes. Moist heat control. I don't want to recite the table. Okay. Remember, there's a table there. This one, sorry. This one. See the table? Okay, so do you need to know the conditions? No, God forbid. Example uses. Again, be reasonable. Say, I don't remember what were the practice exam questions. Okay, surgical room. Can you spray it with a peracetic acid? You can. Can you guarantee that everything is clean? No, you cannot, because there are many nooks and crevices there. So probably something gaseous that can fill the room and stay there for a while. Does that make sense? Or I can ask you, what about food? You say, I'm going to dump it in the bleach. Well, sure. It's not going to help though. I'm going to throw it in the autoclave. Nah, it's a pretty bad idea. Food's not going to be edible after that. So, I don't know. Radiation would work. That's perfect. Okay, does that make sense? So, like, be reasonable when we talk about this. Sterilizing, disinfecting, it's all there, it's all on the page. Dry heat control, same story. Bacteriostatic, I'm basically referring to refrigeration and freezing. Radiation control, UV versus gamma, basically. Non-penetrating versus penetrating. So that's range of electromagnetic spectrum. I'm not doing physics here. Okay, so I say UV light, least penetrating power, disinfection. Gamma rays and X-rays, high penetrating power, gamma rays has the highest. Sterilization. Underlying mechanisms, ionizing DNA breaks, non-ionizing thymine dimers, gamma rays sterilizing, UVs disinfecting. Again, usage. Can you use gamma rays to sterilize massive amounts of water? You can, but it's going to cost you dearly, and nobody's going to approve of having massive gamma ray cannon to sterilize water. HEPA filtration, high efficiency particulate air used to clear air. You can use filtration for disinfection of liquids. The advantages and disadvantages. Disadvantages, especially for gases, it's very costly to handle the gas. Advantages, you can sterilize, actually, disinfect and sterilize without affecting the chemical composition of your media or gas. Again, main points of consideration for choosing chemical microbicidal agent. Tincture is the alcohol base. Again, what would you use to, I don't know, wash your bathtub? If you suggest to only use peracetic acid, it's not available for it. It's really toxic, you know. What would you use on the skin of a patient if you suggest you're going to be using 30% hydrogen peroxide? Not a great idea. Okay, now, mechanisms, halogens, oxidation, chlorine, iodine, used for usually disinfection. You can use chlorine for sterilization, but it's hard. So mainly disinfection. Chlorine bleach for disinfection of fomites, iodine in the form of betadine for aseptic techniques. Disadvantages, they're toxic. Iodine can also stain. Its level. Its derivatives, they disrupt membranes and denature proteins. Triclosan was used as the component of the soap. Two reasons why its usage has been stopped. It's not as effective as everybody thought it would be, and bacteria developed a resistance to it. Antimicrobial action of bisbiguanidins, disruption of the membranes, and denaturation of the proteins. Chlorhexidine is commonly used for aseptic techniques, so it's asepsis, and it has a very low toxicity, and this is why it's so convenient. Antimicrobial actions of alcohols, disruption of the membranes, denaturation of the proteins. Ethanol and isopropanol are commonly used for disinfection of the surfaces and aseptic techniques. Disadvantages: a flammable, and sort of a side note, people drink it. That's another reason why. Okay. You should really watch Louis Black feature on vaccines. That was great. Just check it. Peroxides oxidation, hydrogen peroxide and peracetic acid are commonly used. So they can be used for aseptic techniques on the patient's skin in a certain dilution. In high concentrations in the solution form, they can be sterilizing, sporicidal. In the gaseous form, hydrogen peroxide is sterilizing by a hydrogen peroxide vaporizing machine. And peracetic acid in the form of sporclins produced by Steris is a sterilizing chemical. Disadvantages: well, they corrosive, toxic, irritating, bleaching, so it's pretty bad. But yeah, they can do disinfection and sterilization. Surfactants, cationic surfactants. We discussed cationic and anionic. Cationic are more effective because they can bind, they positively charged, they can bind to the negatively charged bacterial membrane, disrupt it. Anionic, simply using sanitization, they remove microorganisms from the surface. Surfactants are usually used in the form of a soap or some liquid. Disadvantages: they're not super effective. Cationic can provide disinfecting level of control, but remember we did antibiotics and antibacterial chemicals lab. Lysol is cationic. Okay. So that's the thing. And yeah, anionic surfactants like soap, only sanitization. Heavy metals inhibit enzymes. I'm not going to ask you which ones do you know. Don't worry about it. Disadvantages: well, you have to cover surfaces. You can't just spray someone with gold. Okay. Disinfecting level of control. Alkylating agents, chemically modified DNA and RNA. DNA and proteins, I'm sorry. Which ones do you know? So we have one big group of alkylating agents. It's beta-propionolactone, ethylene oxide, propylene oxide. And another big group are aldehydes: formaldehyde, glutaraldehyde, phthalaldehyde. So propylene oxide, ethylene oxide are gaseous. Beta-propionolactone is liquid. Formaldehyde is a gas. Phthalaldehyde and glutaraldehyde are liquids again. Have an idea. So what they used for: preservation of specimens. Formaldehyde is used to sterilize the rooms. Ethylene oxide, propylene oxide are used to sterilize plastic goods because they gas. Disadvantage: they all are very toxic. Now, this stuff, you see the questions here? Folks, Table is yours. I'm not going to ask you to memorize the date of discovery. Anyone has a comment? Are we good? Are we fine? These folks, what they did. That's about it. Okay. Oh, okay. How do we tell the level of microbial control? Let me go. Sterilization. This is why I try to, so I mentioned it in the lectures, in the YouTube lectures, but I try to go over it relatively quickly. So disinfection, all of these folks are disinfecting except triclosan, they suck. All are disinfectants, you see. Cationic are disinfection, anionic sanitization, peroxides. If you see sporicidal and sterilization, you got it. Disinfection. So microbicidal basically means disinfection, antiseptic. With a long exposure, chlorine can be sporicidal, but meaning that it's sterilizing, but not really good. So I just listed them, the main levels of control, and same goes here. Okay. We talked about filtration being can be sterilizing, as well as like ionizing radiation. So ionizing radiation, sterilization, UV light disinfection. And that goes for temperature as well, at least it's here. So Perry, did I answer your question? Okay, cool. I'm gonna skip this one. Okay. What type of antibiotics, based on the source? So that's this part. Like natural, derived from nature, synthetic, synthesized from scratch, semi-synthetic, natural antibiotics that are modified. Spectrum of action, broad versus narrow, effect, cidal versus static, administration route. Question, anyone?
Yeah, for that slide.
Good.
For broad, is that just you would use a broad antibiotic if you knew it was gram positive or gram negative?
If you don't know what it is, you're going to use broad spectrum.
If you don't know what it is, okay.
Of course, like the first choice, one of the first choice antibiotics when you walk in UTI is cephalosporins, third generation cephalosporins, because they well tolerated, broad spectrum, very well studied. And doctors will start treating you right away before any results come back. Because, we have to do it now, So that is how it works. And can it be gram positive? I mean, your UTI, well, not your, but a UTI, yes, it can. Can it be gram negative? Yes, it can. So you want to use broad spectrum. If I walk in and show the symptoms of rickettsial infection, Rocky Mountain spotted fever, for instance, I can almost bet my olives that it's going to be doxycycline, okay? Because it's great against gram negative intracellular infections. Am I clear? That's a narrow spectrum. Am I clear?
So it would treat both positive or negative?
Broad spectrum, yeah, that would fully kill everything, yes.
And then for the where it kills, like when you use like a strong bacteria, a strong antibiotic, what's doing to our normal bacteria that we have that you get the C. diff?
Is that a question on the study guide or just in general? It's fine, just in general. So the problem with the broad spectrums is that they just swipe the stuff away. They can considerably alter your microbiome, like in the gut. So here's the illustration of it. So you see you have all kinds of microorganisms in your gut. Then you wipe them out because you use cephalosporins, I know. And what you get in the end, you get a bunch of drug-resistant microorganisms now growing to the massive proportions because they don't have any competition. Did I answer your question, or I answered something else and not your question? Zalba.
No, you answered it. Basically,
Okay. Go ahead.
they become resistant, and then the antibiotics don't work, and then they start growing out of control, I guess.
Well, it's not like, okay, they're not becoming resistant. They are resistant, but we kill all microbes that are non-resistant. Does that make sense? Like here, you see that little pathogen that I'm circling around with the cursor. See it? Why can't it grow to massive numbers? Because everybody else, every other microorganism, competes with it.
Oh, okay.
When you take antibiotics, everything else is dead. No competition anymore. You got it. You got a super infection. That's basically a description of C. diff. Like, seriously, description how people get Clostridium difficile. Okay, like I would guess that if we test our class for C. diff in the stool, we're gonna have some people with Clostridium difficile there. That's totally normal. It's pretty common. Okay, and yeah, if you will treat those people with like massive amounts of antibiotics there, they're high risk. Other than that, they're fine. Did I answer?
Yeah, thank you.
Okay, sure. So yeah, administration, you're choosing, like, if something has a really severe side effects, you probably use it only topically. If something is well tolerated, orally or parenterally, IM, intramuscular IV, need something that would last longer and will hit the highest concentration faster, IV, like getting in the bones, for instance, Antibiotics is synergistic, meaning that either of them is bacteriostatic, but together they're cidal. Classes, folks, I'm not going over. I'm not reading you this table. You can read it yourself. Okay. I'm not trying to sound like an ass, but seriously, we can do it. We'll go over some stuff. So penicillins, they beta-lactam antibiotics, target the formation of peptide links in the cell wall. That's the mechanism. Cellular target is a cell wall. Mechanism of resistance. Enzyme called penicillin modifies penicillins, so they're not effective anymore. Then you can use clavulanic acid, which inhibits penicillinase, and penicillins are effective again. Difference between different drugs, side chains. The importance of the difference, they may have different bioavailability, so parenteral route versus oral administration. It may help to overcome the resistance, different spectrum of action stuff. Cephalosporins, also beta-lactams, same cell wall inhibition of peptide linkages. Different generations, they have different spectrum of action. I'm not asking which one has which. Don't worry about it. Difference, yes, side chains. Monobactams, carbapenems also inhibit the formation of peptide linkages. Brain fart, I'm sorry. Formation of peptide linkages in the cell wall, prevent the formation of the proper cell wall, so cell can lyse. Carbapenems are the best class of antibiotics that we have so far. Broadest spectrum, well tolerated. Bacitracin, It inhibits the transport of the components of the cell wall to the cell wall. Vancomycin prevents the formation of glycan chains themselves and peptide linkages, so again, cell wall. Protein synthesis inhibitors, difference between eukaryotes and bacteria. They have different structures of the ribosomes. So if you inhibit 70S ribosome, the bacterial ribosome doesn't really affect the eukaryotic ribosome. And that's the same reason why antibiotics, these antibiotics, these ones that inhibits the cell wall, this is the reason why cell wall inhibitors are well tolerated. Human cells don't have cell walls. Major classes of protein synthesis inhibitors. Slides, folks. Aminoglycosides lead to the formation of, so they target 30S subunit of the ribosome, leading to the formation of defective proteins, which overwhelm the cell, cellular, endoplasmic, cytoplasmic stress. Tetracyclines target 30S subunit, preventing the interaction of tRNA with the ribosome. Macrolides, lincosamides, and streptogrammins, they prevent the release of the growing chain from the ribosome. So they basically, if the growing polypeptide chain, so if this chain cannot get out, that's it, protein synthesis stopped. Now that's chloramphenicol. It prevents the formation of the peptide link between the incoming amino acid and the growing polypeptide chain. Now, pay attention, these ones, they all target 50S ribosomal subunit right here. Oxazolidinones, they inhibit the formation of the initiation complex right here. So that's your protein synthesis inhibitors. Polymyxin. So I want to a few words about polymyxin just to remind you. Cellular target for polymyxins is the membrane, which makes them superficial and also super toxic. So toxic, so that polymyxin B is used only topically, and colistin is used orally in IV, but it's a last resort antibiotic. So I showed you this bowel decontamination, systemic infection by multiple drug-resistant bacteria, So spectrum of action, don't worry about spectrum of action, but they have high levels of side effects because they destroy the membrane, and membranes in bacteria and humans are similar. Okay. Main clinical applications for polymyxin B, topical administration, colistin, last resort. They're not used systemically. Lipopeptides disrupt membranes. Fluoroquinolones inhibit DNA gyrase, preventing DNA replication. Rifamycin inhibits, brain fart again. RNA synthesis inhibits transcription. Nitrimidazole. So the story about nitrimidazoles here, nitrimidazoles, degradation of DNA, possibly inhibition of DNA repair. To be honest, we have no idea. Okay. Pretty common drug. We don't know how flagell works. It's been used for years, but we don't know exactly how it works. We know that it does something bad with the DNA, and then we see fragments of the DNA, but what exactly happens, we're not sure. Sulfa drugs competitively inhibit two different steps of the same metabolic pathway. It leads to the generation of nucleotides. So no nucleotides are generated. Without nucleotide, cell is not good. Trimethoprim inhibits one step, competitive inhibition. sulfa drugs one step, trimethoprim another step, but when they are together, they become bactericidal, and each one of them is bacteriostatic. So sulfa drugs and trimethoprim inhibit two different steps of the same metabolic pathway. Isoniazid inhibits the synthesis of mycolic acid. Therefore, it's selectively acting on mycobacteria. So that's main clinical application, mycobacteria. And diaryl quinolones, they inhibit ATP synthase, suffocating the cell, basically inhibiting respiration. High levels of side effects because ATP synthesis in bacteria and in humans are relatively similar. Main mechanisms of antibiotic resistance, 7. I will see if I can name them myself. So we've got drug modification. Let's go over those. So we've got drug modification, right? When that's penicillinase. Penicillinase changes the structure of penicillin. Drug is modified, not effective anymore. Target modification. So say fluoroquinolone inhibits DNA gyrase. DNA gyrase changes its structure a little bit. Fluoroquinolone cannot bind it, cannot recognize it. It's no good. So that's target modification. Target overproduction. If sulfa drug inhibits whatever, I don't remember the exact the enzyme that it inhibits some. What's that first one? Dihydropteroid synthetase. So if you overproduce the target enzyme, sure, you can inhibit 10% of it, but 90% of it is still functional. Efflux pump, you can pump the drug out. You can prevent the cellular uptake by reducing the number of channels or proteins that get the drug in. Target reproduction. Enzymatic bypass. So microorganisms kind of develop some other metabolic pathway, and target mimicry. When microorganisms produce a molecule that looks like a target for antibiotic, but not a target, so antibiotic binds to it instead. And then we talked about mini cells, but since we're not in the class, it's a pretty cool story, but I'm not going to overload you. I'm not going to ask about mini cells. Drug-resistant bacteria, okay. Dudettes, I told you a couple of days ago, no numbers, no clinical considerations, mechanism, yeah, it's okay. Just you saw the question. You saw the question. Okay. Bauer disk diffusion test. That's what we've done in the class. It measures the resistance of microorganisms to a specific antibiotic. Advantages, it's cool. That plunger thing, admit it, it was awesome. It's also cheap and fast. Disadvantages, you cannot measure minimal inhibitory concentration. So if you look at this slide here, not this one, you have preset concentrations on those disks, okay? So you can't measure minimal inhibitory concentration, okay? You can using dilution methods. So you dilute, you see the lower the concentration of antibiotic, see the concentrations, here, at 8 micrograms per mil, antibiotic inhibits the growth, at 4 it doesn't. So your minimal inhibitory concentration is 8. Here, see, no. Minimal inhibitory concentration, 0.06. Here, no growth. Minimal inhibitory concentration, 8. Here, still growth. So minimal inhibitory concentration is not even on the plate, right? So that's the idea. Everybody got it? Okay. So same goes for the E-strip or E-test. At this point, there's no inhibition zone, or like here, say, well, that's minimal inhibitory concentration. It inhibits pretty well here, but then it goes down. So that's E-test it's pretty cheap and fast. So like all the dilutions are pre-made for you. Okay. So dilutions, we discussed dilutions advantages. They may be a little bit tedious and you have to make dilutions and stuff, but disadvantages, but advantages, it allows you to measure MIC. So E-strips, dilution tests all allow you to measure MIC. Now, therapeutic index, I don't know if it was complicated or not, but it's calculated this way. So ED50 is median effective dose, meaning drug works in 50% of the patients. Does that make sense? If there are no comments in the chat room, I presume that everybody follows me. So ED50 is median effective dose, 50% of the patients, fine. ED50 is the median toxic dose, which means 50% of the patients experience toxic effects of the drug. Therapeutic index is TD50 over ED50. So say if a drug has a therapeutic dose, effective dose of 1 and toxic dose of 10, what's the therapeutic index? 10 over 1. It gives you 10. Let me write it down, the example. So say ED50 is 2 kilogram body weight. ED50 is 100 . What's the therapeutic index? Give me your answer in the chat room. Or you can unmute you and just tell me. So if median effective dose is 2, median toxic dose is 100, what's the therapeutic index? Come on, don't hesitate. You can write it directly to me if you don't want to write it to everyone. Anyone? So remember, the higher the therapeutic index, the better. So what's the therapeutic? I know that I will, you know that I will write. There you go. Yeah, it's 50. Good job, Nate. Yeah, that's 50. And basically what it tells you, the bigger the gap between TD50 and ED50, the higher the therapeutic index. That's what therapeutic index tells you. You have more wiggle room to increase the dose of the drug. Campylobacter, gram-negative. Not all species are pathogenic. They can be found in the mouth. Scapnophilic, microaerophilic, gram-negative road. Disease, bacterial gastroenteritis. Symptoms, diarrhea and vomiting. Autoimmune condition associated Guillain-Barré syndrome. Transmission route, zoonotic with poultry and milk. That's it. Ebola virus enveloped, negative strand RNA virus. Transmission route, from bats to humans, usually to the bat blood, from human to human bodily fluids. It does not, it respiratory, hemorrhagic fever, multiple bleedings and fever. Geographically, Africa. Main reasons for the Ebola virus outbreaks in Africa: frequent encounters with the wildlife, lack of proper epidemiological surveys and healthcare. Anyone has a question? I've heard a sound. No, we're good? Okay. It better survives on porous surfaces. Blood increases the survival of Ebola virus. That's by the way, that's example. How does blood affect the survival of Ebola virus? It's really cool because that organic compounds in the blood, they protect Ebola virus from being inactivated by air or temperature. Mycobacterium tuberculosis is gram-positive bacteria, which has a layer of mycolic acid on the outside. Is the vaccine for Ebola effective? That's a good question. So far from what we can see, yes. Yes. Okay. Yes. For our intents and purposes, yes, we do have a vaccine and it's effective. Yes. Do we have that question? Oh, yeah. Sorry, I skipped it. I'm sorry. I skipped it. Vaccine. Yes, there's vaccine available. It's effective. And there are treatments. We have treatments. Sorry for skipping. Mycobacterium tuberculosis usually infects macrophages. Mycolic acid provides protection in this process, increasing the survival. Distinct pathological structure that is formed around the cells infected with mycobacterium is granuloma. And the main cause of pathogenesis is the host immune response, not so much bacterium itself. So it's the host immune response and inflammation that leads to the pathogenesis. Mycobacterium tuberculosis lies dormant in the granuloma, but immunosuppression can reactivate it, leading to the spill into the, you might call it lungs. Infection is ridiculously easy to transmit, but the infection causes clinical disease only in 5% of the cases. In the primary tuberculosis, lungs are affected. In the chronic tuberculosis, also lungs are affected. Symptoms of the chronic pulmonary tuberculosis, chest pain, shortness of breath, cough, fatigue, night sweats, low-grade fever, bloody or greenish sputum. In extrapulmonary or miliary tuberculosis, any organ, from brain to heart to kidneys, bones. Prognosis for the disease is poor. Mycobacterium is very prevalent. Main diagnostic tests: sputum stain, chest X-ray, TB test, PCR. Vaccine. BCG vaccine, marginally effective. It is not used in the US for that reason. It is used in other countries to prevent the transmission from between the children and prevent extrapulmonary tuberculosis in children. The best preventative measure is hygiene and prevent overcrowding, where this infection is transmitted very easily. So I'm going to stop recording. That kind of concludes the whole thing, and I'm going to switch back to WebEx.